Thank you for everybody. So I'm going to tell you recent work we have done working on the Varicella Zoster virus in terms of identify the genes required for viral replication, and that study may lead to a potential novel vaccine development. So very brief introduction. So VZV belongs to the herpes virus family. Actually, this morning you just heard about HSV 6 or 7. VZV actually belong to the same family of those two herpes viruses. It contains a linear double-strand DNA and it has 225 kb genome encoded 71 open-ended frames. And there's a clinical strain called POCA, which uses a lot in our laboratory studies, and also this strain actually used for developing the VZV vaccine. So very briefly tell you the VZV caused human diseases. The disease starts from infection and of the mucosal surface, and the virus then infected the dendritic cells and then infect T cells in lymph nodes. The T cells are going to carry the virus to the internal organs for amplification, and then after amplification, the virus actually will infect T cells, and then that infection will cause a skin rash. That's the chickenpox. At the same time, virus is stable latency in the neurons, especially in this dorsal root ganglion. So when people are getting older, immunity getting weaker, the virus could reactivate and then send the virus back to the skin, which will cause herpes zoster, also we call it shingles. So as you know, we have the vaccines developed against these two diseases. First, I want to talk about a little bit herpes zoster because most people before vaccine was introduced had chickenpox, and then during the course their lifetime, the virus could reactivate, and maybe half of 50% of population will suffer the zoster. And some of them are going to suffer with this severe neural pain, which could last a long time. And there's no very specific treatment for that. Of course, we do have the antiviral against VZV infection. The reason they cause severe pain compared to the herpes simplex virus is simplex virus reactivation only cause local infection in the sensory neurons and then cause local replication in skin. But in the case of VZV reactivation, the reactivation could spread to the other neurons in the ganglion and cause severe infection and damage of neurons. That results in the pain. So I mentioned the vaccine. Vaccine against chickenpox was introduced in 1995. It's pretty effective. And recently, zoster virus actually was also approved. Basically, they are same vaccine. It's called the VOCA strain. So the differences between these two is just because this zoster virus vaccine have about 14 times higher titer than chickenpox vaccine. And there are some potential problems for this vaccine. First, this vaccine actually is mixed strain. That means just a virus from clinical strain pass in vitro in non-human cells many times. So virus, this strain is attenuated, it's a live attenuated virus, but they still have ability to infect the neurons. And there is some evidence reported that this actually latent virus could reactivate or may cause shingles. So this is now very safe vaccine. And also because the titer is very low, the production of this vaccine is difficult, especially more difficult for production of a Zoster vaccine. It requires a much higher titer. And of course, it's very difficult to manipulate this virus because it's a mixed strain, and it cannot be used as the viral vector, as you just heard. So our goal is to develop a next generation of VZV vaccine, which have both skin and neural attenuated, and also genetic defined, easily manipulate. Hopefully, we also can modify the virus so that we will have a higher yield. So when we start this project, we look the viral genome. This is a large DNA virus. This is much more complicated, and more than half of genes has not ever been studied. The reason for that is first, the VZV only infect humans. There's no animal model for this to study this virus. And secondly, the virus is highly associated, cell-associated. It's difficult to make recombinant viruses. It's extremely difficult using the conventional method like co-transfection and recombination and selection the recombinant viruses. So the first thing we did is to generate a VZV bacterial artificial chromosome, basically cloned complete viral genome into E. coli so that we can use bacterial genetics to produce recombinant viruses. So basically we insert one viral vector, bacterial vector, into back vector into one of the cosmate of these four overlapping cosmate and co-transfect into the human cells. So homologous recombination occurred and then produced the viruses. So because there is a GFP marker there, so that the plaque is green. And also this vector contains the chlorophenical resistant gene. So when we isolated this large circular DNA from E. coli, from infected cells and transfecting to E. coli, then we select for the full-length DNA in E. coli. So when we isolate DNA from E. coli, then this virus DNA is infectious and can generate a virus which is same as the original parental virus. And then we further modified this by insertion another inserting another marker, which is a luciferase gene, so that we can later monitor viral replication both in vitro and in vivo using this in vivo imaging system. So once we have this system, we study generate recombinant viruses by systematically delete each viral opening frames. Okay, and generate recombinant virus. So the library of recombinant viruses with each opening frame is deleted. So to summarize this work, we found 44 opening frames are essential, and the 24 opening frames are not essential for viral replication in vitro. And among them, actually, 18 of opening frames did not show any phenotype, growth phenotype defects in vitro. So our next thing is to just focus on these 18 recount viruses and see which gene is required for viral replication in skin and neurons. So first thing we did is screen for the requirement for infection of skin. So we use this in vitro skin organ culture, okay, from fetal tissue, okay, and then inoculate virus, okay, each of these in the skin, and after at different times we measure the luciferase activity as shown here. So the parental virus show the infection, okay, you see a lot of bioluminescence light. There's one mutant to show great defects. And then basically by this screening, we found 4 of these opening frames mutant have a severe defect in the skin, including opening frame 7, that's a newly identified skin tropic factor. Okay, we made a rescue virus and show the virus, once we put white type in, virus can grow normally. Okay, so the next thing we tried neuroblastoma to see any of these mutants can infect these neurons. This just show you the parental viral infection. Yeah. So this study actually showed, surprisingly, the only one opening frame is required for replication in the neurons, which is OFF7. Okay. Here just show you results. So this virus can deletion, we call 7D, can grow in the epithelium cells, but not neuron. But rescue virus can. Okay, so by this study, we identified essential and non-essential genes. Especially, I think OFF7 is most interesting opening frame because single opening frame deletion will cause defect infection of skin and neurons. So that could be a good candidate for the vaccine development. Of course, those studies are in vitro, and more importantly, to show their function in vivo. And since we don't have the animal model, and then so we use this ski mice with implanted fetal tissues. Basically, we use, in this case, the thymus liver from human and implant in the kidney under the kidney capsule, and after 2 to 3 months, this grow like a thymus-like tissue, and then we can inoculate the virus and then monitor viral replication. So here, luciferase imaging here will help us a lot to see viral replication in vivo. So actually, this is the first time to see it in vivo replication of VZV. So we first look for the infection of T cells, right? In the ski mice with thymus implants, because remember the vaccine for vaccine live attenuated, we need virus infect T cells and burn virus in vivo in internal tissues for amplification. So here to show you there's no significant differences between parental virus and 7D deletion virus in T cells. For the skin, we use the same system, just implant the fetal skin under the mouse skin. Okay, so basically there are 2 pieces of skin for each mice, for mouse, and then you see parental virus can replicate very well, and the mutant 7 deletion mutant fail to replicate in skin. For the neuron, here we implant the dorsal root ganglion under the kidney capsule. So this is the right tissue is human dorsal root ganglion. Again, inoculate the virus, we can see 7 deletion also have a significant defect in neurons. and now we want to see the whole infection pathway. So here we use the infected dendritic cell, okay, labeled as red by virus. Okay, so here to show you the infected dendritic cell can carry the virus and infect ARP cells or neurons. Okay, so dendritic cell can be infected by 7D. And then we use that dendritic cells actually inject simers, and then you can see both 7D also rescue the virus, and then they can infect T cells. And then we actually isolate the T cell from this infected tissue and inject in the mouse with skin implants, okay, from the tail wing. Okay, the infected cells actually can carry the virus to infect the skin. This is Y type, but 7D is failed. We did the same experiment for the neurons. Basically, we did some functional assays I'm not going to talk to here. So just to summarize some of our results, some of them I didn't tell you, don't have time. So basically 7D can replicate in different types of cells in vitro, especially dendritic cells and T cells, but it's attenuating skin and neurons. And then actually we know it's localizing Golgi and associated with surveillance. So also it interacts with some other viral proteins. So now just summarize. So we do find this 7D is skin and neural attenuated strain, so genetically defined, easily manipulated. Right now the yield is still not higher. We're still working on it. So I want to just finish up by discussing. So we did hear the viral vectors. So I think in addition, this will be, I think potentially will be safer vaccine for chickenpox, and also could be a good vector, a viral vector for other vaccines, because it's first is safe. We know that chickenpox vaccine has been used many years, and they will cause very good immunogenicity, and the DNA virus very stable, and also it can actually, because the genome can carry very large piece of foreign DNA, okay, and potentially that could be another interesting viral vector. Okay, I will stop here. So I will thank people who did the work and my collaborators.